Welcome to our channel. We are so glad you found your way here. Let us help you fall asleep. Let's begin with extremophiles, the bacteria that eat radiation and acid. Take a slow, deep breath in and release it. Let the weight of the day fade away as we embark on an unhurried, atmospheric journey through the wonders of the cosmos. Your mind is quiet. The universe is vast, and we are ready to explore. Extremophiles represent organisms that thrive in conditions considered inhospitable to most life forms. These remarkable entities exist in environments characterized by extreme temperatures, acidity, salinity, and radiation. Their resilience is not merely a testament to life's adaptability, but serves as a critical focus of scientific inquiry. Studying these organisms enhances our understanding of the fundamental principles of biology and the limits of life on Earth. This region of study reveals the biochemical mechanisms enabling life to flourish under such harsh conditions. For instance, specific extremophiles have evolved unique proteins and enzymes that function optimally at temperatures exceeding 100 degrees Celsius. Others possess cellular structures that withstand high levels of acidity or radiation, allowing them to survive where conventional organisms would perish. The significance of extremophiles extends beyond Earth. Insights gained from these organisms inform astrobiology, the exploration of potential life on other planets, and the conditions that may support it. They challenge preconceived notions of what constitutes a habitable environment. Understanding their metabolic pathways can also lead to biotechnological applications, including waste management and bioremediation processes. The existence of extremophiles invites profound questions regarding the nature of life, adaptation, and the potential for life beyond our planet, reshaping scientific perspectives and expanding the boundaries of exploration in astrobiology. Microbial life represents a diverse spectrum of organisms, ranging from simple prokaryotes to more complex eukaryotes. Bacteria, as the most abundant life forms on Earth, exhibit remarkable adaptability, thriving in environments previously thought inhospitable. These single-celled organisms possess unique characteristics, including rapid reproduction and genetic exchange mechanisms, allowing for swift evolutionary responses to environmental changes. This region is also home to archaea, another type of prokaryote, which often inhabit extreme conditions similar to those favored by extremophiles. Their unique biochemical pathways allow them to harness energy from sulfur and methane, contributing to biogeochemical cycles that sustain ecosystems. Eukaryotes, characterized by their more complex cellular structures, include organisms such as fungi, protists, and multicellular plants and animals. The evolution from simple prokaryotic organisms to complex eukaryotic life marks a significant milestone in the history of life on Earth. This transition was facilitated by endosymbiotic events, where certain bacteria became integral components of eukaryotic cells, leading to the development of organelles like mitochondria and chloroplasts. The study of microbial life not only provides insights into the resilience and adaptability of life forms, but also raises questions about the potential for similar organisms existing elsewhere in the universe. The implications for astrobiology remain profound, inviting continued exploration of life's boundaries. The discovery of extremophiles transformed the understanding of life's resilience, In the 1970s, researchers began identifying organisms thriving in seemingly inhospitable environments. This era marked a significant shift in the perception of life, challenging long-held beliefs about the requirements for survival. Initial findings in hot springs revealed that certain thermophiles could endure temperatures exceeding 100 degrees Celsius. Subsequent explorations into acidic environments, such as those found in Yellowstone National Park, unveiled acidophiles capable of thriving in pH levels comparable to battery acid. These organisms demonstrated that life could not only exist in extremes, but flourish, expanding the known thresholds of biological endurance. The discovery of halophiles, which thrive in high salinity environments like salt flats and salt mines, further illustrated the adaptability of life. The findings prompted a re-evaluation of where life might be found, extending the search beyond Earth. In 1983, a groundbreaking moment occurred when scientists detected microorganisms in a deep-sea hydrothermal vent. These communities existed in complete darkness, fueled by chemosynthesis rather than photosynthesis, challenging the notion that sunlight was essential for life. Each milestone revealed a new layer of complexity, emphasizing the need for deeper inquiry into the resilience of life. The exploration of these environments continues to inform not only biology but also the search for extraterrestrial life. The existence of extremophiles offers profound implications for astrobiology, particularly in understanding the potential for life beyond Earth. These remarkable organisms thrive in conditions previously thought to be uninhabitable, such as extreme temperatures, high acidity, and intense radiation. Such resilience suggests that life may not be limited to Earth-like environments. This region of study expands the horizons of habitability across the cosmos. Research into extremophiles reveals metabolic pathways capable of utilizing unconventional energy sources. For instance, certain bacteria utilize radiation as a food source, transforming harmful rays into chemical energy. This adaptability proposes that similar life forms could exist on other planets or moons, particularly those with harsh conditions, like Europa or Mars. The implications extend to the identification of potential biosignatures, which may not resemble traditional life forms, but could indicate the presence of these resilient organisms. The search for extremophiles deepens our understanding of life's mechanisms, opening up new avenues for exploration. Future missions may target environments previously dismissed as too hostile. The study of these microorganisms transforms our comprehension of life's diversity, suggesting that the universe may be teeming with uncharted forms of existence. By examining these resilient life forms, scientists continue to unravel the complexities of biology and the potential for life beyond our planet. Diverse classifications of extremophiles reveal the remarkable adaptability of life. These organisms are often categorized based on their preferred environments, showcasing their unique characteristics. Thermophiles thrive in high-temperature settings, such as geothermal vents. They possess specialized proteins that maintain stability under extreme heat, enabling metabolic processes that would be impossible for typical life forms. Psychrophiles, in contrast, flourish in frigid conditions, often found in polar ice or deep ocean waters. Their cellular structures are adapted to remain fluid and functional at temperatures well below freezing. This adaptability allows them to metabolize and reproduce in environments that would be lethal to most organisms. Halophiles prefer hypersaline environments, such as salt flats and salt mines. They have evolved mechanisms to balance osmotic pressure, allowing them to survive in conditions that would desiccate other life forms. Acidophiles inhabit highly acidic environments, such as acidic hot springs or mine drainage. Their cellular components are uniquely structured to withstand low pH levels, enabling metabolic activity where few others can endure. Alkalophiles thrive in basic conditions, commonly found in soda lakes. Their enzymatic processes operate optimally in high pH environments, showcasing the vast range of conditions under which life can persist. Each type of extremophile illustrates the remarkable resilience and adaptability inherent in biological systems, expanding our understanding of life's potential across the cosmos. Radiation-resistant bacteria exemplify some of the most extraordinary adaptations found within extremophiles. One prominent species, Deinococcus radiodurans, possesses a unique capacity to withstand high levels of ionizing radiation. Its remarkable ability to repair DNA damage allows it to survive doses of radiation that would be lethal to most other organisms. This bacterium's resilience stems from its highly efficient DNA repair mechanisms, which include multiple copies of its genetic material, providing redundancy that aids in recovery from radiation-induced damage. Another notable organism, Rubrobacter zelenophilus, thrives in extreme environments characterized by both high radiation and desiccation. This bacterium's structural adaptations enable it to persist in conditions that would typically obliterate cellular integrity. It showcases the diverse strategies life employs to endure in inhospitable settings. The study of these organisms has implications beyond understanding life's limits. Insights gleaned from their biology offer potential applications in bioremediation, where their capabilities might be harnessed to clean environments contaminated by radioactive waste. Research into their genetic and metabolic pathways continues to reveal mechanisms that could be applied in biotechnology and medicine. In the broader context of astrobiology, these resilient species provide clues about possible life forms on other planets, particularly those exposed to high radiation environments, suggesting that life's tenacity may extend far beyond Earth. Mechanisms of radiation resistance in bacteria involve intricate biochemical pathways and unique cellular structures. These microorganisms often possess extraordinary DNA repair systems, allowing them to correct damage caused by ionizing radiation. Enzymatic processes, such as the activity of specific nucleases and polymerases, play critical roles in recognizing and repairing double-strand breaks in genetic material. In many cases, proteins like REC-A facilitate homologous recombination, a vital repair mechanism. This process not only restores DNA integrity, but also enables genetic diversity, enhancing adaptability to extreme conditions. Additionally, some extremophiles produce pigments, such as melanin, which absorb harmful radiation and mitigate cellular damage. Intercellular antioxidants, including superoxide dismutase and catalase, help neutralize reactive oxygen species generated by radiation exposure, further protecting vital cellular components. The presence of specialized membrane structures may also contribute to their resilience, acting as barriers that limit the penetration of damaging agents. In environments rich in radioactive materials, These survival strategies allow bacteria to thrive where most life forms would perish. The understanding of these mechanisms not only deepens comprehension of fundamental biological processes, but also opens avenues for innovative applications in fields such as waste management and space exploration. Insights gained from their resilience may one day facilitate the development of novel approaches to enhance human health and environmental sustainability. The ability of extremophiles to thrive in environments saturated with radiation stems largely from their sophisticated DNA repair mechanisms. These organisms possess unique proteins and enzymes that detect and rectify the molecular damage inflicted by ionizing radiation. This process often involves multiple pathways, such as nucleotide excision repair, homologous recombination, and non-homologous end joining. The efficiency of these pathways allows for rapid recovery, ensuring survival in conditions that would be hostile to most life forms. In this region of extreme conditions, certain extremophiles exhibit remarkable adaptations that enhance their DNA repair capabilities. For instance, some utilize specialized proteins that stabilize their DNA structure, protecting it from the destabilizing effects of radiations. Additionally, the presence of pigments such as carotenoids can absorb harmful radiations, further shielding the cellular machinery. These adaptations not only ensure the survival of extremophiles, but also provide valuable insights into the fundamental processes of life. Understanding the intricacies of DNA repair in such organisms may offer clues to enhancing similar mechanisms in human cells. This research can inform approaches to combat aging, cancer, and genetic disorders. As scientists delve deeper into these biological marvels, the potential applications expand, highlighting the interconnectedness of life in both terrestrial and extraterrestrial environments. Biofilms play a crucial role in the survival of extremophiles, particularly in environments rich in radiations. These complex aggregations of microorganisms create a protective matrix that enhances resilience against harmful radiations. The structural composition of biofilms, primarily composed of polysaccharides, proteins, and nucleic acids, acts as a shielding barrier. This layer absorbs and deflects radiation, mitigating its impact on the sensitive cellular machinery contained within. Within these biofilms, communities of bacteria engage in various interactions, facilitating nutrient exchange and waste management that bolster their collective survival. The dense clustering of cells within this matrix fosters a microenvironment where conditions can be finely tuned, offering protection from both high acidity and radiations. Metabolic pathways in these communities adapt, allowing for enhanced efficiency in energy production. This resilience is not merely a survival strategy, but also an evolutionary advantage. Biofilms can withstand extreme fluctuations in environmental conditions, allowing extremophiles to thrive in the harshest of habitats on Earth. These adaptations could also provide insights into the possibilities of life in similar extraterrestrial conditions, where radiation levels may be pronounced. Understanding the mechanisms of biofilm formation and function could lead to advancements in bioremediation and bioengineering, highlighting the remarkable capabilities of life to endure and adapt. Acidophiles thrive in environments characterized by low pH levels, often found in acid mine drainage, sulfuric hot springs, and volcanic craters. These organisms have evolved to flourish in conditions that would be inhospitable to most life forms. The extreme acidity of their habitats forces them to adopt unique metabolic pathways, allowing them to utilize alternative energy sources and sustain their existence in environments with high concentrations of hydrogen ions. This region of the biosphere plays a critical role in various biogeochemical cycles, Acidophiles contribute to mineral weathering and nutrient cycling through their metabolic processes, transforming their harsh surroundings into habitats that can support other organisms. The ability of these bacteria to thrive in such conditions is a testament to life's resilience, showcasing adaptive strategies that enable survival under extreme duress. For the fascinating interactions between acidophiles and their environments extend to their potential applications in biotechnology. Their resilience to acidity can be harnessed for industrial processes, such as metal recovery and bioremediation of contaminated sites. Understanding these organisms not only provides insights into microbial life, but also opens avenues for innovative solutions to environmental challenges. The study of acidophiles highlights the remarkable versatility of life, revealing the intricate balance between organisms and the extreme environments they inhabit. Acidophilic bacteria exhibit a unique biochemistry that enables survival in environments characterized by low pH levels. This adaptation involves specialized cellular structures and metabolic pathways that facilitate growth in highly acidic conditions. The membranes of these microorganisms contain distinct lipids that maintain stability and integrity, preventing damage from the corrosive acidity surrounding them. Enzymes produced by acidophiles demonstrate exceptional functionality under extreme pH, often retaining their activity in conditions where most life forms would succumb. These enzymes play crucial roles in metabolic processes, allowing for efficient energy extraction from substrates that would be toxic to other organisms. The pathways utilized by these bacteria often involve the oxidation of metals and sulfur compounds, further illustrating their ability to thrive in environments replete with heavy metals and other toxic elements. The genetic makeup of acidophilic bacteria is equally fascinating. Gene expression is finely tuned to respond to pH fluctuations, enabling rapid adaptation to varying environmental conditions. Some species possess plasmids that confer additional resistance to acidity and toxic metals, showcasing the evolutionary strategies employed to survive and flourish in hostile habitats. This remarkable biochemistry not only provides insights into the resilience of life, but also holds potential applications in biotechnology and environmental remediation, where these organisms may contribute to bioremediation efforts in contaminated sites. Maintaining pH homeostasis is crucial for the survival of extremophiles in highly acidic environments. These organisms utilize specialized mechanisms to regulate their internal pH, ensuring cellular functions remain uninterrupted. One primary strategy involves the use of proton pumps, which extrude excess protons from the cell. This process effectively counteracts the influx of protons from the external environment, preserving a stable internal pH. Additionally, some extremophiles employ the synthesis of unique proteins that act as molecular chaperones. These proteins stabilize cellular structures and enzymatic functions under acidic conditions. The presence of these adaptations enables organisms to thrive where most others would perish. Intercellular buffering systems also play a vital role in acid resistance. Compounds such as amino acids and polyamines can neutralize excess acidity, providing another layer of protection. This buffering capacity allows for greater flexibility in metabolic processes, even when external conditions fluctuate dramatically. Some extremophiles possess the ability to alter their membrane composition, incorporating specific lipids that enhance membrane stability in acidic surroundings. This adaptive flexibility is a key aspect of their survival strategy, allowing them to withstand extreme pH levels while maintaining essential biological functions. Through these sophisticated mechanisms, extremophiles offer profound insights into the limits of life and the potential for future biotechnological advancements. Extremophiles play a crucial role in biogeochemical cycles, contributing to the cycling of essential elements within diverse ecosystems. Their ability to thrive in environments characterized by extreme conditions enables them to participate in processes that would otherwise be impossible. In acidic or alkaline conditions, these organisms facilitate the breakdown of organic matter, thereby influencing nutrient availability. This region, often overlooked in conventional studies, supports microbial communities that drive the transformation of nitrogen and sulfur compounds. These transformations are vital for maintaining soil fertility and regulating atmospheric gases. For instance, certain extremophiles are involved in the nitrification process, converting ammonia into nitrate, which is a more accessible form of nitrogen for plants. The presence of these organisms in extreme environments, such as hydrothermal vents and salt flats, contributes significantly to the global carbon cycle. They assist in carbon fixation, capturing carbon dioxide and converting it into organic molecules, which are foundational for sustaining various life forms. The resilience of extremophiles underpins their ecological significance, as they serve as indicators of environmental health and stability. Their metabolic pathways provide valuable models for understanding biochemical processes that operate under extreme conditions. Research into these microorganisms continues to unveil their potential applications in bioremediation and industrial processes. The potential of these microorganisms extends significantly into the realm of bioremediation, particularly concerning radioactive waste. Many extremophiles thrive in environments saturated with radiation, utilizing unique metabolic pathways to harness energy from toxic substances. This ability positions them as critical agents in the degradation of hazardous materials. In specific regions, these resilient bacteria can be introduced into contaminated sites with elevated radiation levels. They metabolize complex compounds, transforming them into less harmful substances through biochemical processes. Their presence not only mitigates environmental hazards, but also enhances the safety of areas previously deemed too dangerous for human intervention. Field studies have demonstrated that certain extremophiles possess the capability to absorb and sequester radioactive isotopes, incorporating them into their cellular structures. This process effectively reduces the bioavailability of these isotopes, minimizing their impact on surrounding ecosystems. The resulting byproducts are often far less toxic than their original forms. Research has also identified specific strains that exhibit remarkable tolerance to extreme acidity, allowing them to thrive in conditions that would otherwise inhibit most life forms. This dual resilience provides an invaluable resource for industries seeking sustainable methods to manage hazardous waste. Continued exploration into these microorganisms promises to refine techniques for addressing the challenges posed by radioactive contaminants, offering a glimpse into a safer, cleaner future. Deinococcus radiodurans, often referred to as Conan the bacterium, showcases remarkable resilience against extreme environments. This microorganism can withstand ionizing radiation levels up to 5,000 Gray, a dose lethal to most forms of life. Studies indicate its extraordinary ability to repair DNA damage, a process that involves complex protein interactions and efficient repair mechanisms. Surprisingly, the bacterium thrives in conditions that would be inhospitable to many organisms. It can endure desiccation, extreme temperatures, and high concentrations of toxic substances. This robustness allows it to inhabit environments such as nuclear reactors and radioactive waste sites, where few others can survive. The cellular structure of this extremophile contributes to its durability. A thick peptidoglycan layer shields the delicate inner components from radiation. Additionally, unique proteins play a critical role in stabilizing DNA during damage, and facilitating rapid repair. Research into its metabolic pathways reveals a remarkable versatility. This organism can utilize various carbon sources, providing insights into potential applications in bioremediation. Its inherent ability to metabolize organic compounds in harsh environments could lead to innovative strategies for detoxifying contaminated sites. Understanding Deinococcus radiodurans not only sheds light on the resilience of life, but also informs future microbial applications in environmental science and biotechnology. Genetic adaptations of this organism exemplify the profound capabilities of life to thrive under extreme conditions. It possesses a unique genome structure, sized by multiple copies of its chromosomes, allowing for redundancy in genetic material. This redundancy plays a critical role in its ability to repair DNA damage, particularly from ionizing radiation. Within its cellular machinery, specialized proteins detect and respond to DNA breaks, facilitating efficient repair pathways that maintain genomic integrity even after exposure to high levels of radiation. Additionally, the organism synthesizes a suite of protective enzymes and antioxidants that mitigate the effects of oxidative stress. These adaptations enable it to neutralize harmful reactive oxygen species generated during radiation exposure. The expression of genes involved in DNA repair, such as those encoding for various recombination proteins, is upregulated in response to environmental challenges. In acidic environments, certain genes confer resistance by producing protective biofilms that shield the cells from harsh conditions. These adaptations highlight an evolutionary trajectory that has fine-tuned its survival mechanisms over millennia. The implications of studying these genetic traits extend to bioremediation efforts, where harnessing such resilient properties could lead to novel applications in cleaning up contaminated environments. This organism stands as a testament to the versatility and durability of life in the face of adversity. Genomic studies reveal a fascinating landscape of adaptations among radiation-resistant bacteria. These microorganisms possess unique genetic features that enable survival in extreme environments. Specific genes, known for their roles in DNA repair mechanisms, are highly conserved across various species. This highlights a remarkable evolutionary strategy, allowing them to efficiently mitigate damage caused by ionizing radiation. The comparative genomics of these resilient organisms uncovers a wealth of information. Sequence analysis indicates that certain proteins involved in oxidative stress response are overrepresented. These proteins function to neutralize harmful byproducts generated during radiation exposure. In addition, variations in metabolic pathways suggest a diverse range of energy sources, including the utilization of inorganic compounds. Some bacteria exhibit enhanced capabilities for repairing double-strand breaks in their DNA, a critical factor in their survival. These adaptations not only confer resistance to radiation, but also allow them to thrive in environments characterized by extreme acidity and desiccation. Phylogenetic studies reveal intriguing evolutionary relationships among these extremophiles. By tracing genetic lineages, researchers can uncover how adaptations have emerged over time in response to environmental pressures. These insights contribute to a deeper understanding of life's resilience on Earth and potentially beyond, as similar mechanisms may be found in extraterrestrial environments. The implications extend into biotechnology, where lessons drawn from these organisms may inform novel applications in fields such as bioremediation and medicine. The evolution of extremophiles often hinges on horizontal gene transfer, a mechanism that allows genetic material to be exchanged between organisms, accelerating adaptability. This process enables them to acquire traits that would otherwise take countless generations to develop in environments characterized by extreme radiation or acidity. The ability to swiftly obtain resistance genes can mean the difference between survival and extinction. Bacteria, in particular, are adept at this form of genetic exchange. They can share plasmids, small DNA molecules that can carry advantageous traits with neighboring cells. This sharing occurs through several methods, including transformation, transduction, and conjugation. Such mechanisms facilitate rapid responses to changing environmental conditions, enhancing their resilience in hostile habitats. The prevalence of horizontal gene transfer among extremophiles leads to a rich genetic diversity, contributing to their robust evolutionary success in acidic hot springs. For instance, species may acquire genes that confer metal resistance, enabling them to thrive in environments laden with toxic elements. The interconnectedness of these organisms through gene sharing fosters a dynamic ecosystem where evolutionary pressures are met with swift genetic innovations. In the context of environmental resilience, asizim thu ticris, asizim thu f anitokopsize edawidynaments asizimingsuj. This adaptability underscores the potential of extremophiles as models for understanding how life can persist under the most challenging conditions. Their genetic fluidity continues to intrigue researchers, exploring the limits of life on Earth and beyond. The remarkable adaptations of psychrophiles allow them to thrive in frigid environments where standard life forms struggle to survive. These microorganisms flourish in temperatures often below 0 degrees Celsius, a stark contrast to the conditions that support the majority of Earth's biodiversity. Found in polar ice, deep-sea trenches, and high-altitude glaciers, they possess unique biochemical mechanisms that facilitate metabolic processes despite the oppressive cold. Enzymes produced by psychrophiles exhibit heightened activity at low temperatures, ensuring cellular functions remain efficient even in extreme conditions. This adaptation, known as psychrophilicity, alters the structure of these proteins, resulting in a flexibility that supports life in environments that would otherwise halt biological processes. The membranes of psychrophiles are also modified, featuring higher concentrations of unsaturated fatty acids, which maintain fluidity and functionality in icy surroundings. Research on psychrophiles extends beyond understanding their survival strategies. It also offers insights into potential applications in biotechnology. Their enzymes may be harnessed for industrial processes that require low-temperature operations, promising energy savings and reduced environmental impact. The resilience of these extremophiles serves as a testament to the adaptability of life, revealing possibilities for life in similar extreme environments on other celestial bodies, such as the icy moons of Jupiter and Saturn. Extremophiles thrive in environments characterized by elevated temperatures, such as hot springs and hydrothermal vents. These microorganisms possess unique adaptations that allow them to maintain cellular integrity and functionality in conditions that would be lethal to most life forms. The proteins within thermophiles exhibit remarkable stability, often possessing a greater number of ionic bonds and hydrophobic interactions compared to their mesophilic counterparts. This structural resilience enables them to operate efficiently at temperatures exceeding 100 degrees Celsius. Biochemical pathways in thermophiles are optimized for high-temperature activity. Enzymes, often referred to as thermozymes, exhibit enhanced kinetic properties, facilitating rapid metabolic processes. These adaptations not only support energy production, but also enable the synthesis of complex biomolecules under extreme thermal conditions. Metabolic pathways may include variations of glycolysis and the citric acid cycle, adapted to function effectively in such environments. These organisms also utilize unique membrane lipids composed of saturated fatty acids, providing increased fluidity and stability at high temperatures. The preservation of cellular structure is crucial for survival, allowing them to occupy ecological niches that are inhospitable to other organisms. In understanding thermophiles, researchers glean insights into the potential for life in extreme environments beyond Earth, where similar high-temperature conditions may exist, such as on certain exoplanets or moons. Halophiles thrive in saline environments, where the concentration of salt exceeds that found in typical oceanic settings. These organisms possess unique adaptations that allow them to maintain cellular function amidst high osmotic pressure. Specialized proteins, known as compatible solutes, help stabilize cellular structures and enzymes, ensuring metabolic processes continue despite the surrounding environment's harshness. The mechanisms of adaptation are varied, with some halophiles utilizing salt as a source of energy, while others rely on intricate cellular pumps to regulate internal ion concentrations. These pumps actively transport ions to balance osmotic pressure, allowing the cells to maintain turgor and integrity. Such strategies reveal the remarkable versatility of life, even under conditions that would be detrimental to most known organisms. In locations like salt flats and hypersaline lakes, halophiles contribute to the ecosystem's overall composition, forming microbial mats that can dominate the landscape. Their presence facilitates nutrient cycling, providing a foundation for other life forms to thrive in otherwise uninhabitable regions. The study of these extremophiles not only expands knowledge of life's resilience, but also raises questions about potential habitats on extraterrestrial bodies, such as Europa or Enceladus, where saline environments may harbor similar life forms waiting to be discovered. The deep biosphere, a realm beneath the Earth's surface, reveals the remarkable adaptability of extremophiles. Here, in the absence of sunlight, these microorganisms thrive in environments characterized by extreme pressure, temperatures, and nutrient scarcity. The interplay of geological processes and biological systems creates a unique habitat that sustains life far removed from the familiar ecosystems found on the surface. Chemical reactions occurring within these depths often rely on minerals and gases emanating from geological formations. These extremophiles utilize these inorganic compounds, including hydrogen sulfide and methane, as energy sources. This chemosynthetic activity supports complex communities that function independently of solar energy, showcasing an alternative pathway of life. The implications of the deep biosphere extend beyond terrestrial limits. Understanding these organisms enhances insights into biogeochemical cycles, particularly concerning carbon and sulfur. It also informs astrobiological models by suggesting that similar life forms may exist in subsurface environments on other celestial bodies, where conditions mimic those found deep within the Earth. Research on these extremophiles continues to unveil their significance in maintaining ecosystem stability and nutrient cycling. This ongoing exploration provides a profound appreciation for the tenacity of life, illuminating pathways through which it can endure even in the most inhospitable conditions. The lessons learned from this hidden world inform our understanding of life's potential throughout the universe. Bioenergy production from extremophiles presents a compelling frontier in both scientific research and practical application. These resilient organisms thrive in environments previously thought too inhospitable for life, such as acidic hot springs and radioactive waste sites. Their unique metabolic pathways allow them to convert extreme conditions into energy, offering new possibilities for sustainable energy sources. In this region, extremophiles exhibit remarkable capabilities in breaking down organic matter, utilizing unconventional substrates to produce biofuels. This process not only demonstrates their versatility, but also highlights the potential for harnessing their energy production in biotechnological applications. Companies and researchers are exploring ways to cultivate these microorganisms, aiming to scale their bioenergy production for broader use. Challenges persist, particularly in the cultivation and genetic manipulation of extremophiles. Many of these organisms grow slowly and require specific environmental conditions that can be difficult to replicate. Additionally, the stability and efficiency of biofuel production remain areas of focus. Optimizing these processes could lead to significant advancements in renewable energy. Research continues to delve into the genetic basis of extremophiles, aiming to unlock their full potential. The insights gained may pave the way for innovative biotechnologies that could transform energy production, contributing to a more sustainable future, while deepening our understanding of life's adaptability in extreme environments. Industrial applications of extremophiles in biotechnology are gaining attention for their potential to revolutionize various sectors. Enzymes derived from these resilient organisms exhibit remarkable stability and activity under conditions that would denature conventional proteins. This unique property enables their use in industrial processes that require high temperatures, extreme pH levels, or the presence of heavy metals. In the realm of waste management, extremophiles have been harnessed to bioremediate contaminated environments, effectively breaking down hazardous substances such as heavy metals and radioisotopes. Summon Hedi Galm can tree afford. Extremophilic microorganisms can efficiently convert biomass into energy-rich compounds, often under conditions unsuitable for standard microbial processes. This capability not only enhances yield but also reduces costs associated with energy production. Applications extend to food processing as well, where extremophiles serve as catalysts in the production of fermented products, providing enhanced flavors and improved preservation. Pharmaceutical industries are exploring the genetic material of extremophiles for novel antibiotic and antiviral compounds. Such discoveries could lead to breakthroughs in medicine, particularly in an era of rising antibiotic resistance. Additionally, advances in genetic engineering are allowing scientists to modify these organisms to produce valuable products, expanding their utility further. The ongoing research into these remarkable entities continues to illuminate their importance in addressing some of the most pressing challenges faced by humanity today. The evolutionary history of extremophiles reveals a remarkable tale of adaptation and resilience. These organisms have existed for billions of years, emerging long before complex life took shape. They thrived in environments considered inhospitable, such as hydrothermal vents, acid lakes, and nuclear waste sites. The ancient Earth, characterized by volcanic activity and extreme conditions, provided the ideal backdrop for these early forms of life to evolve. Phylogenetic studies have shown that extremophiles are not a singular group, but rather represent a diverse array of lineages. They belong to various domains of life, including bacteria and archaea, each uniquely adapted to their specific environments. The genetic adaptations observed in these microorganisms often involve specialized enzymes that function optimally at high temperatures or in acidic conditions. These adaptations provide insights into the fundamental processes of evolution and the genetic mechanisms that enable life to survive under extreme conditions. The resilience of these organisms has implications beyond understanding Earth's early biosphere. They serve as models for potential extraterrestrial life forms that might exist on other planets or moons where conditions are similarly hostile. Examining these extremophiles also enhances our comprehension of life's boundaries and the potential for life to thrive in environments previously deemed uninhabitable. The evolutionary journey of these organisms continues to shape scientific inquiry and exploration. Evidence from Mars and Europa offers intriguing possibilities regarding the existence of extremophiles beyond Earth. The Martian surface, once a vibrant environment, now presents a harsh landscape where radiation and extreme temperatures prevail. Instruments have detected organic compounds and seasonal methane emissions, suggesting that microbial life could have once thrived in its ancient waters. The presence of briny liquid beneath the surface raises the prospect of extremophiles existing in subsurface aquifers, potentially adapting to the planet's current hostile conditions. Europa, one of Jupiter's moons, captivates researchers with its icy crust concealing a vast ocean beneath. The potential for extremophiles in this subsurface environment is significant, as conditions might parallel those found in Earth's most extreme habitats. Hydrothermal vents on the ocean floor there could provide energy and nutrients, supporting life forms that survive in darkness and high pressure. The plumes of water vapor detected erupting from the moon's surface offer tantalizing samples for study, possibly revealing the presence of microbial life that has adapted to its unique environment. Both celestial bodies inspire a re-evaluation of life's resilience and adaptability. Their exploration underscores humanity's quest to understand the boundaries of life, continually expanding the definition of extremophiles and challenging our perceptions of habitability in the cosmos. The implications of these findings stretch far beyond Earth, inviting further investigation into life's potential across the universe. The search for life in extreme environments relies on a variety of techniques designed to uncover the hidden potential of survival. Sampling methods are critical. Researchers utilize specialized equipment to collect soil, ice, and water from locations such as hydrothermal vents, acidic lakes, and polar ice caps. These samples undergo rigorous analysis in laboratories, revealing the presence of microbial life that thrives under conditions once deemed inhospitable. Molecular techniques are employed to identify extremophiles. Polymerase chain reaction, or PCR, amplifies specific DNA sequences, allowing scientists to detect even minute populations of microorganisms. This genetic profiling aids in understanding their unique adaptations, such as mechanisms for radiation resistance or metabolic processes that enable survival in acidic environments. Remote sensing technologies further enhance exploration. Satellites equipped with spectrometers analyze surface compositions of distant worlds, providing insights into their potential habitability. Ground-based and aerial surveys augment these findings, employing instruments that can detect organic compounds and signs of biological activity. Experimental simulations recreate extreme conditions in controlled settings, allowing researchers to observe how life forms adapt and evolve. Such studies yield valuable knowledge regarding the resilience of life, contributing to the broader search for extraterrestrial organisms. Each method, whether direct or indirect, brings humanity closer to understanding the vast possibilities of life in the cosmos. The diverse capabilities of extremophiles extend into the realm of food production, showcasing their remarkable adaptability. Fermentation processes benefit significantly from the unique characteristics of these resilient microorganisms. They thrive in extreme conditions, often where traditional organisms cannot survive. This resilience allows them to play a crucial role in producing fermented foods, from yogurt to sauerkraut. enhancing flavors and nutritional profiles. Certain extremophiles can tolerate high salt concentrations, making them invaluable in the production of fermented seafood products. Their metabolic activities not only contribute to preservation, but also promote the development of complex flavors. This region of study is expanding, with researchers exploring the potential of extremophiles to ferment unusual substrates, including waste products from agriculture and industry. In addition to traditional fermentation, these microorganisms offer insights into innovative food technologies. Genetic modifications derived from extremophiles enable the development of enzymes that enhance food processing efficiency. This application can lead to reduced waste and a more sustainable food system. Beyond fermentation, the potential for extremophiles to contribute to the production of biofuels and alternative proteins is under investigation. The ability to utilize non-traditional substrates hints at new pathways for sustainable food sources, ensuring that this remarkable microbial group continues to impact various sectors of food production. Potential medical applications arise from extremophiles, particularly their ability to produce unique antibiotics and enzymes. These microorganisms thrive in environments where conventional life forms struggle, suggesting that their biochemical processes might reveal novel compounds with therapeutic benefits. Insights into their genetic makeup lead researchers to investigate how these organisms synthesize bioactive molecules under extreme conditions. Antibiotics derived from these resilient species exhibit unique mechanisms of action. Their efficacy against multi-drug-resistant pathogens is under examination, providing hope for new treatments in an era where resistance is a growing concern. The specificity and potency of these compounds could revolutionize the approach to infectious diseases, offering alternatives that circumvent existing limitations. Enzymes produced by extremophiles function optimally at extreme temperatures and pH levels. These enzymes, known as extremozymes, are invaluable in industrial processes ranging from pharmaceuticals to biofuels. Their stability and efficiency under harsh conditions make them suitable for applications where standard enzymes would degrade. Research into these biocatalysts continues, revealing potential for enhancing reaction rates and reducing energy consumption. This microbial group exemplifies the intersection of biology and technology, paving the way for innovations in health and industry. Continued exploration of their properties could yield valuable insights, challenging traditional paradigms and expanding the boundaries of scientific knowledge. The implications for medicine and biotechnology underscore the significance of these remarkable organisms. Applications extend beyond medicine into environmental monitoring, where extremophiles serve as innovative biosensors. These organisms exhibit unique survival mechanisms that allow them to thrive in conditions typically deemed hostile, such as extreme radiation or high acidity. Their inherent resilience enables them to act as indicators of environmental health, responding to changes in their surroundings with measurable biochemical reactions. The utilization of extremophiles in biosensing involves harnessing their natural properties to detect pollutants and toxins. In sites contaminated with heavy metals or radioactive materials, these organisms can reveal the presence of harmful substances through alterations in their metabolic processes. Such responses offer real-time data regarding environmental quality, providing insights into remediation efforts and ecosystem stability. Research has shown that specific strains of extremophiles can be engineered to produce fluorescent markers when exposed to contaminants. This bioluminescence serves as a visual cue for monitoring hazardous conditions. As monitoring technologies advance, the integration of these organisms into sensor systems becomes increasingly feasible, enabling widespread applications in diverse environments. Through these innovations, Extremophiles not only deepen our understanding of life's adaptability, but also contribute practical solutions for sustainable environmental management. Their presence in harsh ecosystems continues to reshape methodologies in monitoring and preserving the delicate balance of our planet's environment. Climate change introduces significant challenges to the habitats of extremophiles. Alterations in temperature patterns, ocean acidification, and shifts in precipitation affect the stability of these unique environments. These microorganisms, resilient though they may be, rely on specific conditions for survival. When fluctuations occur, their ecosystems face an uncertain future. Thermophiles thrive in hot springs, where elevated temperatures create a suitable niche. Rising heat from global warming poses a risk to these sites, potentially pushing temperature thresholds beyond what these organisms can endure. Similarly, acidophiles, which flourish in acid-rich environments, may find their habitats altered by increased runoff from changing weather patterns, leading to unpredictable pH levels. In polar regions, psychrophiles inhabit icy landscapes, where melting glaciers expose them to new conditions. The rapid loss of ice cover alters the microbial communities, potentially allowing non-native species to invade. This disruption may lead to a decline in extremophiles that have adapted over millennia to survive in extreme cold. The implications of these changes extend beyond the organisms themselves. Extremophiles contribute to biogeochemical cycles, influencing nutrient availability and ecosystem health. Their response to climate change could serve as an early indicator of broader ecological shifts, highlighting the interconnectedness of life in even the harshest environments. The microbial loop functions as a critical component in ecosystem dynamics, particularly within extreme environments. Extremophiles play a pivotal role in recycling nutrients and facilitating energy transfer through these ecological networks. They occupy niches where traditional organisms struggle to survive, thriving in conditions of high radiation, extreme acidity, or elevated temperatures. In this region, the ability to metabolize inorganic compounds allows for the conversion of available energy into forms usable by other organisms. For instance, certain extremophiles utilize sulfur or iron as energy sources, effectively supporting a range of microbial communities. These interactions contribute to the stability and resilience of the ecosystem, even in the face of environmental stresses. The metabolic processes of extremophiles not only sustain their populations, but also support higher trophic levels. The organic matter produced through their activities forms the foundation for a food web that extends to more complex organisms. This dependency highlights the significance of extremophiles in maintaining the integrity of their environments. As scientists explore these intricate relationships, they uncover the potential applications for biotechnology and environmental management. Understanding extremophiles could yield insights into bioremediation strategies and sustainable practices in agriculture. Their resilience and adaptability present a model for survival that might inform approaches to global challenges. The study of these remarkable organisms continues to reveal the complexity of life in extreme conditions. Ecosystem engineers play a crucial role in shaping their environments, particularly in harsh settings where conventional life struggles to survive. Extremophiles, through their unique metabolic processes, have the ability to alter pH levels, nutrient availability, and even the physical structure of their habitats. In acidic environments, certain bacteria produce compounds that neutralize acidity, creating more hospitable zones for other organisms. This alteration fosters a cascade of biological interactions, benefiting not just the extremophiles themselves, but also a diverse range of microbial and macroorganisms. In high radiation zones, some extremophiles harness radiation as an energy source, subsequently transforming the surrounding ecosystem. The byproducts of their metabolic activities can lead to the accumulation of essential nutrients, promoting the growth of other life forms. The presence of these microorganisms can create microhabitats, which serve as refuges for more complex organisms, including other bacteria, archaea, and even small invertebrates. These modifications contribute to a unique resilience within ecosystems, illustrating a form of life that thrives in adversity and, in turn, supports others. Over time, the cumulative effects of their activity can lead to significant changes in the landscape, demonstrating the interconnectedness of life even in the most inhospitable conditions. Such dynamics underscore the extraordinary adaptability of life and its capacity to influence and transform its surroundings. Interactions between extremophiles and other microbial communities reveal a complex web of relationships that sustain life in some of the planet's most inhospitable environments. These organisms often coexist with various species, forming intricate networks that enhance their survival. In extreme conditions such as those found in hydrothermal vents or acidic hot springs, extremophiles engage in mutualistic relationships. Cooperation provides essential nutrients and energy, facilitating the overall stability of these communities. Through metabolic exchanges, certain extremophiles can assist neighboring microbes in accessing otherwise unavailable resources. For instance, sulfur-oxidizing bacteria may convert toxic sulfur compounds into usable forms, benefiting both themselves and other organisms in the vicinity. This interplay fosters resilience, enabling microbial communities to thrive despite harsh conditions. Competition also plays a significant role in these ecosystems. Some extremophiles produce antimicrobial substances to inhibit the growth of competitors, ensuring their survival in limited niches. The balance of cooperation and competition contributes to the intricate dynamics of these microbial ecosystems. Such interactions not only highlight the resilience of life, but also illuminate potential applications in biotechnology and bioremediation. Understanding these relationships may lead to innovative strategies for harnessing extremophiles in cleaning up contaminated environments or developing sustainable energy sources, showcasing the remarkable potential of life even in the most extreme circumstances. The role of extremophiles in soil fertility is increasingly recognized as a crucial element in ecosystem health. These resilient microorganisms can thrive in harsh conditions, including those characterized by high salinity, extreme temperatures, or acidic pH levels. Within this challenging environment, they play a significant part in nutrient cycling, breaking down organic matter and facilitating the release of essential nutrients that plants require for growth. Some extremophiles possess the ability to solubilize nutrients, effectively transforming them into bioavailable forms. This process is particularly vital in soils that have been degraded or rendered less fertile due to human activity or climate change. Through their metabolic activities, these microorganisms contribute to the overall soil structure, enhancing its porosity and water retention capacity. As a result, they promote a more sustainable agricultural practice by reducing the need for chemical fertilizers. In addition, extremophiles exhibit a unique capacity for bioremediation, aiding in the detoxification of contaminated soils. Their metabolic pathways allow them to neutralize heavy metals and other pollutants, contributing to the restoration of soil health. This resilience not only highlights the adaptability of life in extreme environments, but also underscores the importance of extremophiles in fostering fertile, sustainable land that can support diverse ecosystems and agricultural productivity. Exploring the biochemical pathways of radiation metabolism, reveals complex mechanisms, enabling these organisms to thrive in hostile conditions. These pathways often involve specialized proteins and enzymes that facilitate the breakdown of ionizing radiation into usable energy. By harnessing radiation, certain bacteria convert it into chemical energy, a process that occurs through sophisticated electron transport chains. Within these microorganisms, DNA repair mechanisms exhibit remarkable efficiency, allowing them to withstand and recover from damage caused by radiation exposure. This capability involves intricate systems of nucleotide excision repair and homologous recombination, which ensure cellular integrity and functionality despite environmental stresses. In addition to radiation, the metabolic processes of extremophiles often include the oxidation of heavy metals and the utilization of sulfur compounds. These pathways not only contribute to energy production, but also play a crucial role in biogeochemical cycling. Through these activities, extremophiles can transform toxic elements into less harmful forms, thereby enhancing the ecological balance in their respective environments. A closer examination of these metabolic pathways indicates their potential applications in bioremediation, where they can assist in detoxifying contaminated sites by leveraging the unique characteristics of these organisms. Scientists can develop innovative solutions to address environmental challenges, further demonstrating the invaluable role of extremophiles in our ecosystem. Synthetic biology presents a realm of possibilities where the capabilities of extremophiles can be harnessed to engineer solutions for pressing issues. Innovations in genetic engineering enable the modification of these resilient organisms to enhance their natural abilities. This process allows for the creation of microorganisms that can effectively break down pollutants or convert waste into valuable resources. In this pursuit, researchers explore the metabolic pathways of extremophiles, unlocking secrets that could revolutionize waste management and renewable energy production. The robustness of these organisms under extreme conditions serves as a model for designing new biotechnological applications. The ability of certain extremophiles to thrive in high radiation or acidic environments also opens avenues for bioremediation in contaminated sites, where traditional methods often falter. By employing engineered strains, environments laden with toxic substances can begin to recover, restoring ecological balance. Future applications may extend beyond environmental remediation. The potential integration of extremophiles into agriculture presents exciting opportunities to develop crops capable of withstanding harsh climates and poor soil conditions. These enhancements could lead to sustainable food production, vital in addressing global food security challenges. As research continues, the intersection of synthetic biology and extremophiles may redefine our approach to ecological preservation and innovation, demonstrating the profound adaptability of life. The origins of life remain one of the most profound mysteries in science, with extremophiles offering intriguing insights into this enigma. These remarkable organisms thrive in conditions previously thought inhospitable, such as extreme heat, acidity, and radiation. Their existence suggests that life may have arisen in environments similar to those where extremophiles now flourish. Theories propose that early Earth was characterized by volcanic activity and acidic lakes, conditions that mirror those inhabited by today's extremophiles. Research indicates that the ability to withstand such harsh environments might have been crucial for the earliest forms of life, allowing them to endure and evolve. Some hypotheses suggest that life could have originated in hydrothermal vents, where heat and mineral-rich waters create ideal conditions for microbial communities. These habitats could have provided the necessary energy and nutrients for primitive organisms to emerge. The study of extremophiles also extends to the possibility of life beyond Earth. Locations such as the icy moons of Jupiter and Saturn present environments that might harbor similar organisms. The resilience of extremophiles expands the potential for life to exist in the universe, prompting scientists to explore astrobiological implications. By examining these organisms, researchers gain a deeper understanding of life's fundamental requirements and the adaptive mechanisms that facilitate survival in extreme conditions. Environmental extremes serve as powerful catalysts in the evolutionary process. Within the harsh landscapes of high radiation or extreme acidity, organisms are subjected to intense selective pressures. These conditions prompt rapid adaptations, leading to unique biochemical pathways and survival strategies. The organisms that thrive here often develop specialized proteins and enzymes, enabling them to metabolize substances that would be toxic or lethal to others. In these environments, the competition for resources is fierce. It fosters not only resilience, but also innovation in metabolic processes. For instance, certain bacteria utilize metal ions as electron donors, transforming what would be considered hazardous materials into essential fuel for growth. This ability to exploit otherwise inhospitable conditions highlights the flexibility of life. Wadu would be wasp or lunas of dunosavte. These traits may include enhanced DNA repair mechanisms that counteract damage from radiation or acid. Over time, the accumulation of these adaptations can result in entirely new species, illustrating the dynamic nature of evolution in extreme environments. Such evolutionary paths can provide insights into the potential for life beyond Earth. Understanding how organisms manage to flourish in these conditions enhances our search for extraterrestrial life and informs our comprehension of life's resilience. The study of extremophiles thus opens windows into both our planet's history and the broader universe. These remarkable organisms possess unique biophysical properties that allow them to thrive in conditions hostile to most life forms. Extremophilic proteins exhibit exceptional resilience, maintaining structural integrity under extreme temperatures, pressures, and pH levels. The stability of these proteins is largely attributed to their unique amino acid compositions, which often include increased proportions of hydrophobic and charged residues. This arrangement enhances their ability to form robust three-dimensional structures, ensuring functional efficacy even in challenging environments. In environments where radiation levels are high, certain extremophiles produce specialized proteins that can repair DNA damage caused by ionizing radiation. These proteins employ intricate mechanisms to recognize and mend damaged DNA strands, thereby ensuring cellular survival. The capability of these microorganisms to withstand and even utilize ionizing radiation is of profound interest, not just for understanding life's limits, but also for potential biotechnological applications. Acidophilic extremophiles thrive in environments with pH levels far below 7, where they produce enzymes that facilitate biochemical reactions in acidic conditions. These enzymes exhibit remarkable catalytic efficiency, making them valuable for industrial processes that require high acidity, such as metal extraction and bioremediation. Research continues to uncover these proteins' intricate structures and functions, revealing insights that may inspire advancements in medicine and environmental science. The resilience of extremophiles demonstrates nature's remarkable adaptability, offering a glimpse into the potential for life in the universe's most inhospitable regions. Unfuswearcease expands the potential for life to exist in the universe, presents challenges that push biological systems to their limits. In this context, extremophiles thrive in conditions synonymous with pollution and climate change, showcasing not only endurance but also an unexpected capacity for innovation. Microbial life. such as certain species of bacteria and archaea, showcases adaptability to high temperatures, acidity, and even radiation. Their metabolic pathways often employ unusual substrates, converting harmful chemicals into less toxic forms. This ability to transform environments indicates a fundamental resilience within microbial ecosystems. Even as human-generated disturbances proliferate, this resilience prompts further exploration of potential applications. Bioremediation, utilizing extremophiles to detoxify contaminated environments, emerges as a practical approach to mitigating industrial waste and pollution. Such strategies could harness the natural strengths of these organisms, promoting ecosystem recovery amidst degradation. Investigations into the genetic adaptations of these extremophiles reveal insights into evolutionary processes, shedding light on potential mechanisms of survival under extreme stress. Understanding these adaptations not only informs ecological balance here on Earth, but may also guide the search for life in environments beyond our planet. The study of extremophiles, therefore, serves as both a reflection of our current challenges and a beacon of hope for resilience in the face of adversity. The role of extremophiles in carbon sequestration reveals a remarkable adaptability to their environments. Certain species thrive in extreme conditions, such as high acidity or radiation, yet simultaneously contribute to the stabilization of carbon compounds. These microorganisms exhibit unique metabolic pathways that allow them to convert carbon dioxide into more stable forms, effectively removing it from the atmosphere. In this process, extremophiles utilize carbon as a primary energy source, thereby transforming it into organic matter. The presence of extremophilic bacteria in environments like acidic hot springs or deep-sea hydrothermal vents indicates their potential utility in mitigating climate change. Their metabolic activities not only foster the creation of biomass, but also enhance the mineralization of carbonates, sequestering carbon in geological formations. Investigations into these microorganisms provide insights into natural carbon cycling processes. The resilience of extremophiles under harsh conditions offers a model for biotechnological applications aimed at enhancing carbon capture and storage. By harnessing their capabilities, researchers can explore innovative strategies to address escalating atmospheric carbon levels. The discovery of extremophiles in diverse ecosystems emphasizes their ecological significance. Understanding their complex interactions within these environments can reveal pathways to more sustainable practices in carbon management, illustrating the potential of life in the most inhospitable places to contribute to the planet's overall health. The search for novel biochemicals within extremophiles represents a frontier in bioprospecting, where researchers delve into environments that challenge the limits of life. These organisms thrive in conditions of high radiation, extreme acidity, and temperatures that would denature typical proteins. Their unique adaptations have led to the production of extraordinary biomolecules, offering potential applications in medicine, industry, and environmental sustainability. This region, where life persists against overwhelming odds, becomes a treasure trove for scientists seeking new enzymes, antibiotics, and metabolic pathways. The resilience of these microorganisms allows them to produce compounds that can function in extreme conditions, which are often stable and efficient under varied temperatures and pH levels. Such properties are particularly valuable in industrial processes that require catalysts capable of withstanding harsh environments. The impact of these discoveries extends beyond mere curiosity, as they can lead to breakthroughs in biotechnology. Enzymes isolated from these extremophiles may enhance biofuel production, improve waste management techniques, and contribute to the development of biodegradable materials. By studying the biochemical mechanisms that allow these organisms to flourish, researchers can harness their potential for innovative solutions to pressing global challenges. In this ongoing exploration, the hidden capabilities of life reveal themselves, transforming our understanding of biological resilience and its prospective applications. The influence of extremophiles extends into the evolutionary narrative of eukaryotic cells, marking a significant chapter in the story of life on Earth. These resilient organisms, thriving in extreme conditions, provide insights into the mechanisms that may have facilitated the transition from simple prokaryotic life forms to complex eukaryotic structures. Genetic exchanges between extremophiles and early eukaryotes likely played a pivotal role in this evolutionary leap, introducing new metabolic capabilities and adaptive traits. Research indicates that the endosymbiotic theory, which posits that eukaryotic cells arose from symbiotic relationships between different species, may have been informed by interactions with extremophiles. This region of evolutionary development showcases how harsh environments can forge unique adaptations and foster genetic diversity. Metabolic pathways found in these organisms, such as those for energy production and stress tolerance, have been integrated into the eukaryotic lineage, enhancing survival and adaptability. The implications extend beyond mere survival. They suggest that the traits honed in extremophiles may have contributed to the rise of multicellularity and complex ecosystems. Insights gained from these remarkable organisms continue to shape our understanding of life's resilience and adaptability. Their existence invites further inquiry into how life can persist and evolve under conditions once deemed inhospitable. This ongoing research enriches the narrative of biology and evolution, offering a deeper comprehension of the interconnectedness of life on our planet. The extremophile paradigm shift in microbiology redefines the boundaries of life itself. Organisms thriving in extreme conditions challenge conventional definitions, revealing resilience previously unimagined. These microorganisms, capable of enduring environments marked by radiation, acidity, and extreme temperatures, prompt a re-evaluation of ecological niches. Their presence in locations such as deep-sea hydrothermal vents and highly acidic hot springs illustrates an unexpected adaptability. Research indicates that extremophiles possess unique biochemical pathways, enabling them to metabolize substances toxic to most life forms. This remarkable adaptation raises questions about the potential for life beyond Earth, particularly in environments once deemed inhospitable. The study of these organisms extends beyond Earth-bound implications, influencing fields such as astrobiology and planetary science. In laboratories, scientists leverage extremophiles for biotechnology applications, exploring their potential in bioremediation and industrial processes. The metabolic capabilities of these organisms may provide solutions for environmental challenges, including waste management and energy production. The implications of this research resonate throughout microbiology, encouraging a more inclusive understanding of life. This evolving knowledge base emphasizes the importance of adaptability, suggesting that life may persist in forms and places previously overlooked. Each discovery invites further exploration, expanding the parameters of life's resilience and complexity across diverse ecosystems. Research into extremophiles raises significant bioethical considerations, particularly in relation to the manipulation of these organisms. The ability to thrive in extreme conditions presents opportunities for genetic modification, pushing the boundaries of what constitutes life. This region of inquiry necessitates a careful examination of the potential consequences on ecosystems, both terrestrial and extraterrestrial. The implications of introducing genetically modified extremophiles into natural habitats warrant scrutiny. These microorganisms possess unique properties that could disrupt existing ecological balances if released without regulation. The potential for unintended consequences on biodiversity or the emergence of new pathogens cannot be overlooked. Ethical frameworks must be developed to address these risks, ensuring that scientific advancement does not come at the expense of ecological stability. Furthermore, the exploration of extremophiles on other planets introduces a host of ethical dilemmas. The possibility of contaminating extraterrestrial environments with Earth-based organisms raises questions about the rights of these alien ecosystems. Protecting potential life forms on other planets is as crucial as understanding those that inhabit Earth. The ethical stewardship of both known and unknown life forms necessitates a commitment to responsible research practices. The exploration of extremophiles has significantly influenced science fiction and popular media, imbuing narratives with a sense of wonder and possibility. Works of fiction often draw inspiration from these resilient organisms, portraying them as otherworldly entities capable of thriving in environments considered inhospitable to life. Such depictions challenge conventional boundaries of biology and stimulate the imagination regarding extraterrestrial life forms. Films and literature frequently showcase these organisms as symbols of hope or survival, illustrating humanity's quest to understand life's potential across the cosmos. In stories, they often serve as a bridge between the familiar and the alien, allowing creators to explore themes of adaptation and resilience. These narratives reflect the scientific understanding that life can emerge under extreme conditions, inspiring audiences to reconsider definitions of existence. Moreover, the mention of extremophiles in popular culture serves to educate the public about the realities of life on Earth and beyond. They provoke curiosity about the vast possibilities of life forms that may inhabit other planets or moons within our solar system. This intrigue fosters a broader appreciation for scientific inquiry and the complex ecosystems that exist in extreme environments. And as research continues to unveil the capabilities of these remarkable organisms, their cultural representation will undoubtedly evolve, further enriching the dialogue between science and fiction. The exploration of extremophiles raises profound questions about the nature of life itself, particularly the concept of life as we do not know it. These organisms thrive in conditions once deemed inhospitable, revealing the adaptability of life. In environments characterized by intense radiation, extreme acidity, or extreme temperatures, they not only survive but flourish, employing unique biochemical pathways. This adaptability suggests that the definition of life may need to be expanded. Traditional parameters, such as those requiring water or moderate temperatures, appear insufficient when considering organisms that metabolize radiation or thrive in sulfuric springs. The complexities of their cellular structures indicate that life may exist in forms and under conditions that remain beyond human comprehension. Research into extremophiles also implies the possibility of extraterrestrial life forms. By studying these unique organisms, scientists gather insights into how life might evolve on other planets or moons with harsh environments, such as the icy crust of Europa or the sulfuric landscapes of Venus. These discoveries challenge preconceived notions of biology and encourage a re-evaluation of what constitutes a habitable environment. The implications extend into various fields, from astrobiology to biotechnology, as the mechanisms that allow extremophiles to thrive offer potential applications in medicine, waste management, and environmental remediation. Laboratories employ specialized methods to culture and study extremophiles, enabling detailed exploration of their unique adaptations. These microorganisms thrive in extreme conditions, necessitating tailored environments to mimic their native habitats. Temperature-controlled incubators maintain precise thermal conditions, while anaerobic chambers eliminate oxygen to accommodate those residing in oxygen-deprived environments. Selective media play a critical role in fostering growth, providing essential nutrients that cater to the specific metabolic needs of extremophiles. This region of microbiology requires extensive knowledge of various culture techniques, including enrichment culturing, where samples from extreme environments are exposed to selective pressures to encourage the proliferation of specific extremophilic organisms. Genomic and proteomic analyses further illuminate their biological processes. The extraction of DNA and RNA allows researchers to examine genetic adaptations that confer resilience to harsh conditions. Advanced sequencing technologies reveal the evolutionary pathways that have shaped these organisms over billions of years. Metagenomic studies assess community dynamics, shedding light on how extremophiles interact with their environments and each other. The study of extremophiles not only enhances understanding of life's resilience, but also inspires innovative applications. Insights gained from these organisms may contribute to breakthroughs in biotechnology, offering solutions to some of humanity's most pressing challenges. These organisms serve as a testament to life's adaptability in the face of adversity. The role of extremophiles in oceanic vent ecosystems reveals a fascinating interplay between life and extreme environments. These deep-sea habitats, characterized by high temperatures and immense pressure, host unique populations that thrive in conditions inhospitable to most forms of life. Hydrothermal vents emit mineral-rich fluids, creating a nutrient-dense environment where extremophiles flourish. These microorganisms, including thermophiles and hyperthermophiles, possess specialized enzymes that allow them to metabolize chemicals such as hydrogen sulfide, found in vent emissions. This process becomes a primary energy source, forming the foundation of an entire ecosystem. As they oxidize these chemicals, they provide organic matter that sustains larger organisms, establishing a complex food web. The presence of extremophiles also influences the geological landscape surrounding the vents. Their metabolic processes contribute to the precipitation of minerals, leading to the formation of unique structures called chimney formations. These structures not only provide habitat, but also serve to alter the local chemistry, impacting the biological communities that inhabit them. The resilience of these organisms in such harsh environments offers valuable insights into potential life on other celestial bodies. Understanding how they thrive under extreme conditions enhances our knowledge of life's possibilities beyond Earth, suggesting that similar extremophiles may exist on other planets or moons within our solar system. The resilience of extremophiles unfolds further when examining their relationship with climate extremes. These microorganisms possess remarkable adaptations that allow them to endure environments characterized by extreme temperatures, pH levels, and radiation in regions experiencing significant climate variability, such as deserts or polar ice caps. Their presence may indicate the limits of life in harsh conditions. Certain extremophiles thrive in hyperthermophilic ecosystems, where temperatures soar beyond 100 degrees Celsius. This heat fosters biochemical processes that yield energy from inorganic compounds, enabling life to flourish in the absence of sunlight. In contrast, psychrophilic extremophiles inhabit frigid climates, showcasing survival mechanisms that protect their cellular structures from ice crystal formation. The influence of acidophiles in acidic environments illustrates another dimension of survival. These bacteria utilize unique metabolic pathways to extract energy from sulfur and iron, existing where most organisms would perish. Such adaptations highlight the intricate interplay between life and extreme conditions. Climate extremes shape not only these microscopic inhabitants, but also the ecosystems they support. Observations suggest that as global temperatures rise, the distribution and behavior of extremophiles may shift, Serving as indicators of environmental change, their ongoing study may provide insights into potential responses of life to future climatic scenarios, revealing the resilience and adaptability inherent within biological systems. Extremophiles thrive in the polar regions, where frigid temperatures and extreme conditions create unique challenges for survival. These microorganisms exhibit remarkable adaptations, allowing them to flourish in environments that would be inhospitable to most forms of life. They possess specialized proteins that maintain functionality in sub-zero temperatures, promoting metabolic processes even when the surrounding environment is frozen. In these icy landscapes, extremophiles play critical ecological roles. They contribute to nutrient cycling, breaking down organic matter and releasing essential elements back into the ecosystem. This process not only sustains their own survival, but also supports other organisms, creating a foundation for biodiversity. The microbial mats formed by these extremophiles can be found beneath the ice, serving as hotspots of biological activity. The ability to withstand high levels of radiation further enhances their ecological significance, as some extremophiles in polar regions can utilize radiation as an energy source. Such capabilities suggest potential applications in biotechnology and astrobiology, where understanding these life forms may inform the search for life on other icy moons and planets. The inherent resilience of these microorganisms reveals a stark contrast to the harshness of their surroundings, demonstrating the intricate balance of life in some of Earth's most extreme environments. The biochemical significance of extremophilic enzymes extends beyond their survival mechanisms. These enzymes, adapted to thrive in extreme conditions, exhibit remarkable stability and activity in high temperatures, acidic environments, or high salinity. This stability renders them invaluable in various industrial applications. Industrial processes often require catalysts that can withstand challenging conditions, which typically lead to degradation in conventional enzymes. The unique properties of extremophilic enzymes allow for greater efficiency in processes such as biofuel production, where high temperatures can enhance reaction rates. The use of these specialized enzymes can reduce costs and energy consumption significantly, presenting a more sustainable approach to energy generation. Similarly, in the field of waste treatment, extremophiles contribute to the breakdown of pollutants under extreme conditions, facilitating cleaner and more efficient waste management solutions. Even in the realm of pharmaceuticals, extremophilic enzymes play a pivotal role. They are utilized in the synthesis of complex molecules, providing pathways that are often unreachable by traditional methods. The ability to function in extreme pH levels and temperatures enhances reaction specificity and yield, proving advantageous in producing drugs and therapeutic compounds. The exploration of extremophiles continues to unveil their potential, offering insights into not only industrial applications, but also the fundamental principles of life itself in environments once deemed inhospitable. Their resilience in harsh conditions provides a unique opportunity for biotechnological advancements related to human health. Certain extremophiles, especially those thriving in acidic or radioactive environments, possess enzymes that exhibit remarkable stability and catalytic efficiency. These enzymes can be harnessed for use in medical diagnostics and therapeutic processes, improving the efficacy of treatments. The study of extremophiles has led to the discovery of novel compounds with potential antibiotic properties. These microorganisms produce metabolites that can inhibit the growth of pathogenic bacteria, offering a promising avenue for combating antibiotic resistance, a growing concern in modern medicine. Individual species may hold the key to developing new antibiotics that can effectively target resistant strains. From to their direct applications in medicine, extremophiles provide a window into understanding the evolution of life on Earth. Their unique adaptations suggest mechanisms by which organisms may survive, not only on our planet, but potentially on other celestial bodies. This knowledge could inform astrobiological research, expanding our comprehension of life's resilience in the universe. Additionally, the metabolic pathways of extremophiles can illuminate cellular processes, leading to advancements in regenerative medicine and biotechnology. Their ability to thrive in extreme environments reshapes perceptions of life's limits and capabilities, revealing the intricacies of survival and adaptation. Genetic engineering of extremophiles introduces innovative avenues for enhancing resilience in various applications. Through precise manipulation of genetic material, researchers harness the unique traits of these organisms, facilitating the development of resilient crops capable of withstanding harsh climatic conditions. This approach involves identifying and isolating specific genes responsible for traits such as drought resistance or elevated tolerance to salinity. Laboratory techniques enable the insertion of these genes into more vulnerable species, promoting an adaptive response to environmental stresses. The resultant hybrids exhibit heightened resilience, allowing them to thrive in regions where traditional crops might fail. These engineered traits extend beyond agriculture. The potential for extremophiles to aid in bioremediation processes becomes apparent. Certain genetically modified strains can break down pollutants in contaminated environments, effectively detoxifying soil and water. This capability highlights the symbiosis between biotechnology and environmental sustainability. The implications of such advancements reach into the realm of medicine as well. Extremophiles possess unique enzymes that function optimally in extreme conditions, providing valuable targets for pharmaceutical development. These enzymes may hold the key to creating more efficient drugs or treatments for a variety of diseases. The exploration of genetic engineering underscores the ongoing quest to understand life's resilience and adaptability, as well as the potential to transfer these attributes to other forms of life, enhancing their survival in a rapidly changing world. The role of extremophiles in coral reef systems reflects a delicate balance within these vibrant ecosystems, found in the most inhospitable environments. These resilient microorganisms contribute significantly to nutrient cycling and energy flow. They thrive in conditions that would be detrimental to most life forms, such as extreme temperatures and salinity variations. This adaptability aids in the survival of coral polyps, which rely on their symbiotic relationships with these microorganisms for sustenance. In nutrient-poor waters, extremophiles participate in the breakdown of organic matter. Their metabolic processes release essential nutrients, supporting not only coral growth, but also the diverse marine life that depends on these habitats. The interplay of extremophiles and corals fosters biodiversity, creating a foundation for complex food webs. As these microorganisms efficiently utilize available resources, they enhance the overall productivity of the reef. The resilience of coral reef systems is increasingly challenged by climate change and ocean acidification. Extremophiles demonstrate remarkable capabilities to withstand these stresses, suggesting potential pathways for adaptation within coral communities. Their presence may offer insights into enhancing coral resilience, promoting recovery from bleaching events. Understanding the ecological importance of these microorganisms within coral reefs is imperative for conservation efforts. Their existence serves as a reminder of life's tenacity and the intricate relationships that sustain marine ecosystems. Collaborations between extremophiles and macroorganisms reveal a complex web of interdependence in extreme environments. In acidic hot springs, these resilient microorganisms thrive alongside larger life forms. They engage in mutualistic relationships, where extremophiles break down toxic substances, making essential nutrients available for nearby organisms. This process allows creatures that would otherwise struggle to survive in harsh conditions to flourish. In the depths of hydrothermal vents, extremophiles continue to play a crucial role. Chemosynthetic bacteria harness chemical energy from the surrounding water, converting it into organic matter. This organic matter becomes the foundation for entire ecosystems, supporting a variety of macroorganisms, including tube worms and giant clams. The presence of these bacteria enables life to thrive in an environment devoid of sunlight. Illustrating nature's adaptability, interactions extend beyond nutrient exchange. Some extremophiles produce metabolites that can protect larger organisms from environmental stresses. This protective mechanism fosters a unique resilience among macroorganisms, allowing them to withstand extreme temperatures and pressures that would otherwise be lethal. The intricate dynamics of these collaborations highlight the vital roles extremophiles play in sustaining ecosystems in seemingly inhospitable environments. Observing these relationships provides vital insights into life's capacity for adaptation, unveiling the underlying principles that govern biodiversity in extreme habitats. Understanding these interactions enriches our knowledge of ecological resilience and the thresholds of life on Earth. Metagenomics provides a powerful tool for examining extremophilic communities, allowing scientists to explore the genetic makeup of diverse microbial populations without the need for culturing individual species. This approach reveals a wealth of information about the functional potential of these organisms, shedding light on their adaptations to extreme environments. In regions characterized by high radiation or acidity, metagenomic studies have identified genes associated with stress response, repair mechanisms, and metabolic pathways that enable survival under harsh conditions. Sampling from various extreme habitats, such as hydrothermal vents or acidic hot springs, has uncovered unique genetic signatures. These signatures manifest specific adaptations, indicating how these microbes utilize available resources to thrive. The ability to analyze genetic data from entire communities rather than isolated strains provides insight into the ecological dynamics at play, including interspecies interactions and competition for resources. Functional metagenomics allows researchers to assess not only what organisms are present, but also the potential functions they serve in their environments. This understanding can lead to discoveries regarding biogeochemical cycles, contributing to broader ecological models. The implications of these findings extend beyond Earth, offering a glimpse into the potential for life on other planets, where conditions may mirror those found in extreme environments on our own planet. As more data becomes available, the complexities of these microbial ecosystems continue to unfold, enhancing our comprehension of life's resilience. The search for alternative energy sources has increasingly turned toward extremophiles, particularly those thriving in harsh environments. These organisms exhibit remarkable metabolic pathways that allow them to convert various substrates into usable energy, showcasing their potential in bioenergy applications. They inhabit extreme conditions, such as high radiation levels, elevated temperatures, and acidic environments, where conventional life forms cannot survive. Various species have been identified that can metabolize elements like sulfur, iron, and even carbon dioxide. This capability opens pathways for harnessing energy production from waste materials. thereby contributing to sustainable practices. For instance, certain extremophiles are being studied for their ability to break down toxic substances while simultaneously generating energy. These unique traits are paving the way for bioremediation efforts, where they can detoxify polluted environments while providing viable energy alternatives. In addition to waste processing, the ability of some extremophiles to produce biofuels is being explored. They can generate hydrogen gas or alcohols through fermentation processes under extreme conditions, presenting a sustainable approach to energy production. Research into these organisms continues to reveal innovative methods for energy extraction, demonstrating how life's adaptability in extreme environments can influence future energy solutions. As investigations progress, the potential of extremophiles as a cornerstone of renewable energy systems becomes increasingly significant. The search for viable habitats beyond Earth hinges on the resilience of extremophiles. Their extraordinary capacity to thrive in extreme environments suggests they could play a pivotal role in the colonization of extraterrestrial landscapes. These microorganisms demonstrate an ability to endure high radiation levels, extreme temperatures, and acidic conditions, making them ideal candidates for life in regions such as Mars or the icy moons of Jupiter and Saturn. In these environments, extremophiles could assist in bioremediation, breaking down toxic compounds found in Martian regolith or other celestial materials. Utilizing their metabolic processes, they may convert harmful substances into usable resources, thereby creating a more suitable environment for human habitation. The prospect of employing extremophiles to produce oxygen or essential nutrients from local materials resonates with the concept of closed-loop life support systems. This region of scientific inquiry extends to the possibility of engineering extremophiles to enhance their capabilities further. Genetic modifications may allow these organisms to perform specific functions, such as synthesizing food or energy from limited resources. Such advancements could pave the way for sustainable life support systems on distant planets, reducing the logistical burden of transporting supplies from Earth. As research continues, the implications of these resilient organisms stretch far beyond Earth, opening a pathway to future exploration and habitation across the cosmos. The microbial ecology of extreme environments showcases a remarkable diversity of life forms, each uniquely adapted to thrive under harsh conditions. Within these niches, organisms exhibit extraordinary metabolic pathways, enabling them to utilize energy sources that would be toxic or inaccessible to most life. In the depths of hydrothermal vents, for instance, chemolitho-autotrophic bacteria harness the energy from hydrogen sulfide, forming the backbone of a complex ecosystem that provides sustenance for various organisms. In acidic environments, such as those found in acid mine drainage, certain bacteria exhibit extreme acidophilicity, maintaining cellular integrity and metabolic function in pH levels that would denature proteins in most living beings. These adaptations highlight the resilience of microbial life in the face of seemingly inhospitable conditions. Saline lakes and arid deserts also provide fertile ground for extremophiles. Halophilic bacteria flourish in high salinity environments, employing unique cellular mechanisms to balance osmotic pressure. The presence of these organisms contributes to the biogeochemical cycles that sustain ecosystem functionality, recycling nutrients and facilitating energy flow. The intricate interactions among extremophiles and their environments offer insights into evolutionary processes, revealing pathways that may inform astrobiology, and the search for life beyond Earth. Understanding these ecosystems can enhance the comprehension of life's resilience and adaptability across the universe. The future of extremophile research holds promise for uncovering new biological mechanisms and applications that could revolutionize various fields. Scientists are increasingly exploring environments that were once deemed inhospitable, such as deep-sea hydrothermal vents, acidic hot springs, and the frozen realms of Antarctica. These investigations reveal that organisms can thrive under extreme conditions, showcasing metabolic pathways and biochemical processes that challenge traditional definitions of life. Recent advancements in genetic sequencing technologies allow researchers to analyze extremophiles at the genomic level, providing insights into their unique adaptations. This deep genetic understanding paves the way for biotechnological applications, such as bioremediation strategies for contaminated environments, or the development of sustainable biofuels. The ability of certain extremophiles to metabolize heavy metals and radioactive materials presents a potential for cleaning polluted sites, addressing pressing ecological concerns. Additionally, astrobiology is becoming increasingly intertwined with extremophile research. The study of life in extreme conditions on Earth serves as a model for potential extraterrestrial ecosystems, guiding the search for life on other planets and moons within our solar system. Missions to Mars and the icy moons of Jupiter and Saturn are informed by the knowledge gleaned from these remarkable organisms. This expanding horizon of extremophile research invites exploration into the very essence of life itself, suggesting that resilience may be a universal trait. The existence of extremophiles reveals an intricate web linking these organisms to global biodiversity. Within the harshest realms of Earth, they thrive alongside more conventional life forms, suggesting a shared lineage. These adaptable microorganisms play crucial roles in nutrient cycling, influencing ecosystem dynamics across various habitats. Their metabolic processes contribute to soil fertility and water purification, underscoring their importance in sustaining life. Terrancing intrication and occurrent, such as hydrothermal vents and acidic hot springs. These organisms demonstrate unique biochemical pathways that allow them to harness energy from otherwise inhospitable surroundings. This ability could provide insights into the potential for life in similar conditions beyond Earth, particularly within icy moons and distant exoplanets. Their resilience offers clues to evolutionary mechanisms that may enhance survival across diverse environments. Interactions between extremophiles and other organisms reveal a complex interdependence. For instance, the symbiotic relationships formed with plants and animals showcase how even the most resilient species can influence broader ecological networks. These interactions highlight the delicate balance necessary for maintaining biodiversity and ecosystem stability. This interconnectedness suggests that understanding extremophiles is imperative for comprehensive ecological studies. Their presence serves as a reminder of the adaptive capacities of life, illuminating the pathways through which resilience permeates the fabric of existence on Earth. Extremophiles not only highlight resilience, but also function as crucial indicators of environmental change. These organisms thrive in harsh conditions such as high radiation, extreme acidity, or elevated temperatures, providing a unique lens through which scientists can monitor ecosystem health. As conditions fluctuate, shifts in extremophile populations may signal broader ecological transformations. In this region, the diversity of extremophiles can reveal subtle changes in climate and habitat. For instance, the emergence or decline of specific species often correlates with alterations in soil chemistry or water availability. This correlation serves as a barometer for environmental stresses, enabling researchers to assess the impacts of pollution or climate change. They persist in some of the most inhospitable environments on Earth, ranging from acidic hot springs to radioactive waste sites. These bacteria not only adapt to their surroundings but also alter them, influencing the biogeochemical cycles within their ecosystems. The metabolic processes of extremophiles can even contribute to bioremediation, breaking down harmful substances and restoring balance. Monitoring these organisms provides critical insights into the resilience and vulnerability of various ecosystems. Their ability to withstand extreme conditions positions them as valuable tools for understanding how life adapts, survives, and transforms in response to the relentless pressures of a changing planet. The exploration of extremophiles has profoundly influenced the discipline of astrobiology. These remarkable organisms, thriving in environments once deemed inhospitable, expand the boundaries of where life may exist beyond Earth. Insights gained from studying their biochemical pathways reveal mechanisms that enable survival in high radiation, extreme acidity, and intense temperatures. Such knowledge informs hypotheses about potential extraterrestrial life forms adapting to similar conditions on planets or moons within our solar system and beyond. Investigations into these resilient microorganisms suggest that celestial bodies like Europa and Enceladus, with their subsurface oceans, might harbor life. The presence of water, combined with the extremophiles' adaptability, supports the idea of thriving ecosystems in environments previously thought barren. The search for biosignatures on exoplanets incorporates these findings, enhancing models that predict the potential for life in diverse atmospheres. The field research continues to uncover new extremophiles, each discovery adding to the understanding of life's resilience. Their metabolic pathways and genetic adaptations serve as biological blueprints for life in extreme environments. This ongoing research not only shapes the methodologies used in astrobiological studies, but also reframes the conceptualization of life's fundamental requirements. The implications of these findings resonate throughout the scientific community, inviting further exploration into the vast possibilities of life's existence across the cosmos. This exploration reveals how extremophiles have shaped the evolution of life on Earth. In the harshest environments, they exhibit remarkable adaptability, enabling them to thrive in conditions that would be lethal to most organisms. Their resilience offers insights into the fundamental principles of survival and genetic diversity. Many extremophiles possess unique biochemical pathways that allow them to metabolize substances such as sulfur or methane, highlighting the vast array of life-sustaining processes that exist in nature. These organisms contribute to biogeochemical cycles, influencing nutrient availability and ecosystem stability. For instance, the presence of extremophiles in extreme environments like hydrothermal vents contributes to the cycling of carbon and sulfur, essential elements for life. This interaction illustrates the intricate web of dependencies that sustain ecosystems, even in the most inhospitable conditions. The study of extremophiles also sheds light on the origins of life itself. Understanding their mechanisms of survival may provide clues about how life emerged on Earth and potentially elsewhere in the universe. By examining the genetic materials of these resilient organisms, researchers can trace evolutionary lineages, offering a glimpse into the past. The presence of extremophiles emphasizes the adaptability of life, suggesting that resilience may be a fundamental characteristic, not only on Earth, but potentially on other planets and moons within our solar system and beyond. These organisms provide crucial insights into the mechanisms of evolution, showcasing how life can endure and thrive in conditions previously deemed inhospitable. Understanding their genetic adaptations illuminates the pathways through which life can evolve under extreme environmental pressures. Each extremophile reveals specific traits that enhance survival, such as unique metabolic processes capable of harnessing energy from radioactive materials or thriving in acidic environments. Such adaptations not only broaden the definition of habitable conditions, but also challenge existing paradigms regarding the origins of life. The evolutionary strategies employed by these microorganisms suggest that life may arise in a variety of forms, even in environments previously considered uninhabitable. This realization extends the potential for discovering life beyond Earth, as similar conditions exist on celestial bodies like Europa or Mars. The study of extremophiles also informs astrobiology, guiding the search for life beyond our planet. Insights gained from these organisms can assist in identifying biosignatures, the chemical indicators of life, in extreme environments within our solar system. This understanding fosters a deeper appreciation for biological resilience and plasticity, highlighting that life may not only survive, but can also flourish under conditions that challenge our conventional understanding of biodiversity. Through this lens, extremophiles stand as a testament to life's remarkable ability to adapt and evolve, setting the stage for future explorations into the universe's potential for life. Innovations in extreme microbiology propel our exploration of these resilient organisms, enhancing our understanding of their unique capabilities. Recent advancements in genetic engineering, for instance, allow researchers to manipulate the DNA of extremophiles, unlocking potential applications in biotechnology and environmental science. By altering specific genes, it becomes possible to enhance their ability to thrive in hostile environments, paving the way for bioremediation efforts in contaminated sites. The advent of next-generation sequencing technologies further accelerates this field. These methods provide unprecedented access to the genetic blueprints of extremophiles, revealing their metabolic pathways and survival strategies. This genetic insight enables scientists to identify and harness the enzymes produced by these microorganisms, which may catalyze reactions under extreme conditions, potentially transforming industrial processes. Additionally, advances in artificial intelligence and machine learning optimize the search for new extremophiles. AI algorithms analyze vast amounts of environmental data, pinpointing locations where these microorganisms may thrive. This targeted approach not only expedites the discovery of novel species, but also enhances our understanding of their ecological roles. These emerging technologies collectively contribute to a deeper comprehension of life's adaptability. As researchers continue to unravel the complexities of extremophiles, they illuminate pathways for harnessing biological processes that could redefine our approach to sustainability and innovation in the face of environmental challenges. The study of extremophiles beckons deeper philosophical inquiries into the very nature of life itself. These organisms thrive in conditions previously deemed inhospitable, raising questions about the boundaries and definitions of life. They exist in environments saturated with radiation, fundifologue, raising questions about the boundaries and definitions of life. They exist in environments saturated with radiation, extreme temperatures, or acidity, challenging preconceived notions of what constitutes a viable habitat. This resilience invites contemplation on the adaptability of life forms, suggesting that life may manifest in forms and conditions beyond human comprehension. In examining these remarkable entities, the potential for life on other planets becomes a focal point. The diversity found within these bacteria implies that extraterrestrial ecosystems might harbor life forms that thrive in environments radically different from Earth. This opens the door to a broader understanding of life's possibilities, positioning extremophiles as critical pieces in the search for extraterrestrial intelligence. Ethical considerations emerge too, particularly in the context of biotechnology. As researchers explore applications derived from extremophiles, questions regarding the manipulation of life for human benefits surface. This intersection of science and ethics necessitates a careful examination of the implications of altering natural processes, emphasizing the responsibility that accompanies such advancements. When contemplating extremophiles, the inquiry extends beyond biology, delving into existential questions about resilience, adaptation, and the essence of life itself. The implications ripple across disciplines, merging science, philosophy, and ethics into a complex dialogue. The future of extremophiles presents an intriguing frontier for exploration, promising insights that could reshuffle the boundaries of current scientific understanding. These resilient organisms thrive in conditions deemed inhospitable, from deep oceanic vents to the harsh landscapes of polar regions. Their ability to metabolize toxic substances, including radiation and acids, holds significant potential for bioremediation efforts, offering natural solutions to environmental degradation. Research into their unique biochemical pathways reveals mechanisms that could inspire innovative technologies, such as advanced materials or sustainable energy sources. Efforts are underway to harness extremophiles in biotechnology, possibly revolutionizing industries ranging from food production to pharmaceuticals. The application of extremophiles might lead to breakthroughs in understanding life's resilience, suggesting that survival mechanisms can extend beyond Earth. Matthew Wackles, as astrobiologists search for life on other planets, these organisms serve as a model for potential extraterrestrial ecosystems. Discoveries may redefine the parameters of life itself, challenging preconceived notions about habitability across the cosmos. The continued study of extremophiles underscores their critical role in both ecological balance and the advancement of human knowledge. Their existence is a reminder of life's tenacity, inspiring a deeper inquiry into the possibilities that lie within the unseen corners of our world and beyond, highlighting the interconnectedness of all living systems.